All right, so today we are going to talk about Chapter 10, Cell Division, and the mini exam for this week will include this chapter and the following Chapter 11. In this picture on the first slide, we can see a cell undergoing division. It's pretty cool looking. One interesting thing about when cells divide in culture is that the two resulting cells will often have a mirror image shape of each other. The objectives for today are to understand how DNA is packaged in the cell. So how is it that such a large amount of DNA can fit into each individual cell? Second, to understand what happens at each stage of the cell cycle. And hopefully this isn't the first time you've learned that, but we'll go over each stage today. The third objective is to be able to describe what happens at each stage of mitosis. We talked about mitosis way back in the second lab of the semester, and so we're going to refresh our memories about what happens at each stage. Something that may be new for you is understanding what cell cycle checkpoints are and how they are controlled. So at what points in the cell cycle are things monitored to make sure that things are going well? And then lastly, since we're talking about cell cycle, when cell cycle is dysregulated, we can have cancer occur. And this dysregulation can be caused by oncogenes and tumor suppressors. So you should be able to understand what these oncogenes and tumor suppressors are. So let's talk about DNA packaging in the cell. So DNA is packaged in chromosomes. The human cells have 23 pairs of chromosomes, or 46 chromosomes in total. Each chromosome can have many genes on them, right? So chromosomes are made up of DNA. And also, in order to be packaged so that it fits into the cell, the DNA is packaged around proteins. And so chromosome includes the DNA and its associated proteins. Now, what you may have heard the phrase or the term chromatin. Chromatin is composed of DNA and the protein that it is packaged with. Right? So chromatin is DNA plus the proteins that package it. One chromosome is actually one continuous, very, very long fiber that's just been packed very tightly into a bundle. Let's look at the breakdown of a chromosome and its structure. So the cross-section of this chromosome, you can see, is composed of a scaffold protein that runs the length of the chromosome, as well as these rosettes, or little petals of like a flower shape, that are made up of chromatin loops. Each chromatin loop is composed of solenoid and the scaffold protein. And now that we get to the solenoid, now we can finally actually see the DNA. And the DNA is wrapped around these proteins called histones. The DNA in association with the histone is called a nucleosome. So the DNA, for simplicity, has just been drawn as a blue strand, but it's a double helix, right? Just like we've learned about DNA. And again, the DNA is wrapped around this protein core called a histone, and the DNA in complex with the histone is called a nucleosome. So it's easy to get histone and nucleosome mixed up, so make sure that you study that. You don't need to memorize some of these numbers, but the histone itself is actually made up of 8 proteins, and one coil of DNA around that histone is about 200 bases in length. Also, if you remember, DNA, the double helix, is made up of sugars and phosphates, a sugar-phosphate backbone. And those phosphates have a negative charge, so they impart a negative charge to the DNA. And the protein that it wraps around, that histone, has side chains, many side chains that have positive charges. And so we have this ionic interaction that promotes the binding of the DNA to that histone. Again, those nucleosomes then coil around each other into a structure called a solenoid. And that solenoid is then formed into a chromatin loop, right? That is formed, the loop structure is formed with another scaffold protein. Now, these chromatin loops and rosettes are only formed during mitosis. You can see it's called a mitotic chromosome. And that is because how tightly DNA is packaged depends on what cell stage it is in. So if you need to divide the DNA, split it in two, as is done during mitosis, it's helpful for the DNA to be packaged very tightly into individual bundles that can be easily separated and sorted. If you imagine a basket of yarn, balls of yarn, if each different color of yarn has been wrapped into its own ball, they're going to be very easily separated. But if, say, you let a cat play with it for a little bit, now it's going to be really hard to organize your yarn by color. And so during mitosis, DNA is packed more tightly. However, when it's not mitosis, this DNA needs to be used, it needs to be accessible in order for gene expression to occur. If you remember, the DNA is used as a template during transcription to make message RNA. That message RNA is turned into protein, and then so you need to have access to the DNA for gene expression to occur. For those polymerases to get in, the double helix needs to be pulled apart. And so when the DNA is more loosely packaged, we call that euchromatin. Here you can see a picture where on one side the DNA is more tightly packaged. The yellow is the histone, and then we have in red the DNA wrapped around. And so you can see that the nucleosomes are tightly packed on the left, right? That is called heterochromatin, and more loosely packaged on the right, and that's called euchromatin. There are also certain cell types that just never need to turn on certain genes, and so those genes will be tightly packed away in the heterochromatin. Here we have on the right a picture. This is called a karyotype. This is an array of all the chromosomes in an organism that have been labeled. And you can see in this example, we're looking at a karyotype for human. There are 23 pairs of chromosomes. In total, 46 chromosomes. And you can see that the 23rd pair, those are the sex chromosomes that determine whether you are male or female. Why do we have two pairs? So when we have pairs of chromosomes, that's called being diploid, right? We have two pairs because one chromosome comes from the sperm and one comes from the egg during initial formation of the zygote. So you have one set one of the pairs is coming from dad and one pair comes from mom. And that is what allows genetic diversity in offspring, right? Our children are not our clones. Now, for a cell to divide, first it has to copy its DNA. Because you imagine if the cell split in two and it didn't duplicate its DNA, now each daughter cell would have half the amount of DNA. And then when each of those daughter cells splits in two, those daughter cells now have a quarter of the amount of the original DNA. And so before the cell can divide, it has to duplicate its DNA, and that occurs during S phase. Sorry, cat's getting into stuff. Here we have a picture on the left of a pair of chromosomes. And a pair of chromosomes they are called homologous chromosomes because they are the same. We've got one homologous chromosome came from egg, one came from sperm. Now, during S phase, these homologous chromosomes are duplicated, right? So each of them gets duplicated. We can see now we have two blue chromosomes and two red chromosomes. And each copy of these duplicated homologous chromosomes is called a sister chromatid. So it's very important that you understand the difference between a homologous chromosome and a sister chromatid. These sister chromatids are kept together by a protein called cohesin at a region of the chromosome called the centrosome. And that is an area of the DNA where it is more constricted near the center. Great, so quiz. Homologous chromosomes and sister chromatids are the same thing, true or false? take a minute, pause the video to think about this question. So again, each pair of homologous chromosomes comes from the gametes. So one comes from the sperm and the other homologous chromosome comes from the egg to form the two. So we have the pair of homologous chromosomes in the zygote. And the sister chromatids only occur during S phase when a cell is going to divide. So if this zygote is going to, say, divide into two, it's going to duplicate that DNA, and each pair is going to be copied, and each copy is called a sister chromatid. So the answer to this question is B. Sister chromatids and homologous chromosomes are not the same thing, and it's important that you know the difference between the two. Let's talk about cell cycle. So the cell cycle is made up of several parts. We have G1 phase, S phase, G2 phase, mitosis, and cytokinesis. Any of the phases that are not mitosis and cytokinesis together are called interphase. And you may have remembered we talked about interphase back in lab 2. Sorry, cat making noise. So G1 stands for GAP1. This is the primary growth phase. So when the new cell is formed, it grows during GAP1 phase. And then eventually it may receive a signal to divide. And so then it will go into S phase. That is where it's going to duplicate its DNA. After synthesis of the DNA, the cell undergoes G2 or gap phase 2. In addition to the DNA being duplicated, organelles also need to be replicated, and the cell is getting ready for division. Mitosis is when the cell splits the DNA into two to each side of the cell, and then during cytokinesis the cell is literally split into two, so the cytoplasm is split into two. And mitosis is further subdivided into 5. Here we have a diagram of the cell cycle. You can see in yellow we have, G1 phase. That is our longest phase of the cell cycle, typically, during which cells grow. And at some point during G1, the cell will receive a signal to divide, and so it will then go into S phase, where it can duplicate its DNA. In red, we have S phase. After the cell's DNA is duplicated, the cell proceeds to G2 phase, shown in green. And then when the cell actually splits its DNA and its cytoplasm in two, we have M phase. So M phase is made up of mitosis, shown in purple, and cytokinesis, shown in blue. And mitosis itself has 5 phases. I encourage you to take a minute to watch this video, especially if it's been a while since you've learned about the cell cycle. I have a copy of these slides on Canvas so that you can click on it and play the video. So how long does all of this take, right? It really depends upon the cell type. For example, during development, the cells are dividing very often, and so the cell cycle is very short so that they can divide sooner. And we talked about that in the lab at the beginning of the semester. We compared different cell types, and we saw that one cell type of a developing fish had a shorter cell cycle, and it was more proliferative. You don't need to memorize any of these numbers. I just wanted to give you a general idea of how long this is taking. For example, M phase, typically in mammalian cells, only takes about an hour. Now there is one unique variation for G1 or GAP1 phase. Some cells, once they are born, never divide again. For example, the majority of the neurons in your brain don't divide. And so these cells, they don't need to divide again. They don't need any of that cell replication machinery. And so they are going to go into something called resting phase, or G0. It's kind of like when you move into a house and you decide, you know what, this is it. We're not moving anymore. And you finally throw away all your cardboard boxes, all right? It's like they're packing up all of that cell machinery stuff, and they're shoving it way under the bed, saying we're not planning on dividing anytime soon, right? So that's what G0 is. Again, interphase is all of the phases except for M phase, so that includes G1, S, and G2 phase. Now, during G2 phase, the cell is getting ready to divide. So the chromosomes, the DNA, is going to start to condense so that it is ready to be easily sorted. And we're going to talk about the different parts of the chromosomes during mitosis, right? So let's look at a picture on the next slide. My cat's a little attention starved. So again, here we have a picture of two sister chromatids. So the cell has undergone S phase and it is replicated its DNA, and so we have two sister chromatids. These sister chromatids, as we said, are held together by cohesin proteins at a region called the centromere. Now, during cell division, during M phase, these two sister chromatids are going to get pulled apart. And so cytoskeletal fibers, microtubule fibers, will be kind of casting out and trying to grab onto the DNA, kind of like you're fishing, right, with a fishing pole. And when they attach to that DNA and pull the DNA apart, they're attaching to the DNA by at this point called the kinetochore. Here we have a picture of the cell during G2 phase. So it's getting ready for M phase. And you can see that the centrioles, these are formed of microtubules, and they are what are going to create the mitotic spindle. So normally during interphase, the cell only has one centriole, but during G2, it's going to copy the centrioles so that there can be one on either side of the cell. During M phase, we have mitosis, which is split into 5 phases. Now, you've heard of prophase, metaphase, anaphase, and telophase before. The way I remembered it was IPMAT, interphase, prophase, metaphase, anaphase, telophase. But some people do further distinguish prophase and split it into 2 parts, prophase and prometaphase, like your book does. So I guess you can remember it as IPMAT. Just do whatever you need to do to memorize these stages. So let's take a minute to do another question. Which of the following phases of the cell cycle are included in interphase? So the answer to this is D, all of the above. G1, S, and G2, anything except for M phase, are all considered interphase. And you can see in this picture, right? G1, S, and G2 all comprise interphase. Anything except for mitosis or M phase is considered interphase. Let's break down those steps of mitosis. Right? Again, we have shown in purple prophase, prometaphase, metaphase, anaphase, and telophase. And then we also have in blue cytokinesis. During prophase, the DNA is going to further condense so that it is easily sorted and split into the 2 sides of the cell. And the mitotic spindle is going to form. So these microtubule fibers that reach out, grab onto the DNA and split those 2 sister chromatids apart. In addition, the nuclear envelope is going to break down because those microtubules that are fishing for the DNA, they are not in the nucleus. And so we need to give those microtubules access to the DNA. You can see that in this picture that the mitotic spindle is beginning to form. These microtubule fibers are starting to grow out from the centrioles, and the DNA in the nucleus, it's tightly packaged and condensed, and the nuclear envelope as well is breaking down so that those green microtubules can access the DNA. During prometaphase, again, this is that phase that some people include in the cell in mitosis. The microtubules that are fishing for DNA are going to capture that DNA. Sorry, I'm making banana bread and it smells really good. And it's going to attach to that DNA at the kinetochore. And then those 2 sister chromatids are going to be pulled back and forth, right? It's kind of like a tug of war going on. Here we can see the microtubule fibers have been casting out and are starting to catch some DNA, and they're playing this tug of war with the DNA. They're attached to the DNA at what? The kinetochore. During metaphase, the DNA, the chromosomes, line up in the middle of the cell. Metaphase, middle. You can see in this picture on the right, they've stained the DNA and the mitotic spindle. The mitotic spindle is in pink. You can see how it's reaching towards the center where the DNA is, and the DNA has been kind of roughly lined up in the center of the cell. And interestingly, where that DNA lines up is where the cell will split in two later on. Again, you can see in this diagram, the mitotic spindle, it's extending from both sides of the cell. The DNA is attached to the microtubules at the kinetochore, and the chromosomes are all lined up in the center. During anaphase, the DNA is going to split apart, anaphase apart. Those two sister chromatids have been attached to each other. You remember by what protein? The protein is called cohesin. Right? Like being cohesive, cohesin. And so that cohesin protein gets degraded, and that allows each sister chromatid to get pulled apart to opposite sides of the cell by the microtubules. Now there are two types of movements of the DNA, or ways that the DNA gets moved to opposite sides of the cell, but I don't expect you to memorize these. But what happens is those microtubules will shorten in length, but then they will also, the centrosomes where they are originating from will also move further and further apart to each other, and together that will cause the sister chromatids to be moved to opposite sides of the cell. As you can see in this diagram, we have each sister chromatid is now separated from each other, and they're being pulled to opposite sides of the cell. Telophase is the last phase of mitosis. The DNA has been successfully split apart onto opposite sides of the cell, and so the spindle is not needed anymore and is going to break down. In addition, the nuclear envelope is going to reform around each little pile of DNA on opposite sides of the cell. And the chromosomes, now that they have been split apart, they don't need to be tightly packaged anymore, and so they are going to start to loosen up. We can see in this picture, we now have two ends of the cell where there is DNA, and the nuclear envelope is going to reform around both sides. We have two nuclei forming, one on each side of the cell, and the mitotic spindle is starting to dissolve and break apart. Now, after mitosis, during M phase, we have cytokinesis, and this is when the majority of the rest of the cell, cytoplasm, is split into two, and you can visually see this under the microscope. And the way that this cell splits in two is that a ring of actin cytoskeleton will form around the cell, and it will tighten and tighten and tighten until the cytoplasm is pinched, and we now have two resulting cells. It's kind of like if I was to take my hair and put it into a ponytail, right? Ponytail is like the actin. Now, I have a video that you can watch if you would like to study up on the phases of mitosis. You can watch this crash course video on YouTube on your own time. Let's take a minute to do a question. What happens during the S phase of the cell cycle? Growth and maturation, replication of nuclear DNA, cell division, or separation of chromosomes into sister chromatids? Right? The correct answer is B, replication of nuclear DNA. Right? So the DNA, the chromosomes are copied, not split in two, they are copied or duplicated, and now we have, for each chromosome, we have a sister chromatid. Let's do another question. The spindle fibers attach to the sister chromatids at the what? So pause the video if you need to. The protein that the sister chromatids attach, that the spindle fibers attach to on the chromatids is called the kinetochore. And we can see in this picture The sister chromatids have been captured by the microtubules, the mitotic spindle, and those microtubules are attached to the DNA via kinetochores. Let's talk about cell cycle checkpoints. Again, this might be new to a lot of you. So it's important for the cell cycle to be regulated. If something goes wrong during S phase, we don't want to keep going on to the G2 and M phase, right? And so the cell monitors itself to make sure that everything is going smoothly. And if it's not, the cell will stop and it won't continue to progress through the cell cycle. So there are 2 points of no return. First is replication of the genetic material. During S phase, when the DNA is duplicated, once it's happened, you can't unduplicate the DNA, right? And then second is the separation of the sister chromatids. Once that duplicated DNA has been split in two to opposite sides of the cell, it's not going to go back again. So there are a couple cell cycle checkpoints, right, that allow the cell to make sure that everything's going correctly, as well as to receive signals when it is time to move on to the next stage. So what actually regulates the cell cycle? We have proteins called cyclins that were first discovered in sea urchins. And they found that the levels of these cyclin proteins would rise and drop and rise and drop. And every time the cyclin levels rose, the cell split into two and then they dropped. And then it rose again and the cell split into two and it dropped. Hence cyclin, cycling, right? So they found that there were a lot of cyclins right at the stage between G1 and S phase. And then another set of cyclins would peak right at the switch between G2 and M phase. So what must happen before each checkpoint is passed, right? So in order for the cells to move on, for example, from G2 in green to M, we have the G2M checkpoint. In order to move on past mitosis into cytokinesis, we have the spindle checkpoint. And to move from G1 phase to S phase, we have the G1S checkpoint. So at the G2M checkpoint, let's think about what has happened so far. During S phase, the DNA was copied, it was duplicated, and during M phase, the DNA is going to be split apart. So it's important for the cell to ensure that the DNA was properly copied. And if not, the cell will not proceed into M phase. Similarly, during the spindle checkpoint, at this point, the DNA, the sister chromatids, have been split and moved to opposite sides of the cell. And the next stage is cytokinesis, in which the rest of the cell will divide in two. And so it's important that the DNA, these chromatids, were correctly separated. And if they were not sorted properly, then the cell will pause and will not proceed on to cytokinesis, and it won't split in two. And then, of course, too, I forgot about the G1S checkpoint, right? So to start everything out, so the cell's hanging out in G1, it's growing, it's doing its functions, receives a signal from outside of the cell. And so at this G1S checkpoint, the cell is saying, okay, we got a signal to divide. Are we done growing? Are we big enough? Do we have everything that we need in order to copy DNA, right? So that's what that G1S checkpoint is. Again, we've got then the G2M checkpoint that makes sure that everything has been copied properly, and the spindle checkpoint in which the cell makes sure that the DNA has been successfully and correctly split to opposite sides of the cell. Now, we talked about these cyclins. Scientists learned that cyclins will rise and fall and rise and fall, and that these cyclins are important in order for the cell to proceed from one stage to the next. So the cyclins have to reach a certain level in order for the cell to leave G2 to get into M phase. And then another set of cyclins must get high enough in concentration in order for the cell to exit G1 and go into S phase, right? Further research has shown that these cyclins work in cooperation with another protein called a cyclin-dependent kinase, or CDK. So its name's not too creative, right? It's a kinase, an enzyme that phosphorylates things that's function is dependent upon binding to cyclins. So once these cyclins are high enough in concentration in the cell, they will bind to these CDKs. So the CDKs are unable to do their job of phosphorylating unless there is enough cyclin present. Once cyclin levels have sufficiently, there's enough cyclin to bind these CDKs, they can activate, and their function in phosphorylating is acting like a switch to turn on all the things that need the machinery that needs to be turned on for the cell to proceed to the next stage of the cycle. Right? So this complex was originally called mitosis promoting factor, or MPF, and your book goes into more of the history of the research of how this was discovered. I don't care that you know too much about it, but what I do care that you know is that these cyclin levels rise and fall and rise and fall. Meanwhile, CDK is just staying at the same state. However, the CDK, doesn't matter how much you have, if it's not bound to cyclin, it can't function. And the CDK activity is critical for the cell to move from one stage of the cell cycle to the next. So CDK levels are always high, but the CDK is not active. Once the cyclin levels rise sufficiently, there's enough of them to bind to the CDKs. Now the CDKs are active and they can turn on the machinery that's needed to proceed through the next stage of the cycle. In addition to needing to bind to a cyclin, these CDKs also need to be phosphorylated themselves. Here we have a nice example of these cyclins that I was talking about. So here we have a cell that's splitting 2 times. So it's going through mitosis in green, then splitting, going into interphase in blue, then it's proceeding to mitosis, the cell splits into 2, and the resulting cells are in interphase again. And so you can see that the cyclin levels rise during interphase, and at their peak, the cell is able to enter into mitosis. After mitosis, those cyclin levels drop. So this specific cyclin is important for proceeding into M phase, mitosis, right? Again, the CDK proteins are present at high levels throughout all the cell cycles, but they are not necessarily active. The MCDKs are only active when they are able to bind to the cyclins. And so only when cyclin concentrations have risen high enough do the MCDKs become active. And when they are active, that allows the cell to proceed through mitosis and divide. Again, let's look at these checkpoints. We have in G1 phase, the cell is hanging out, it's growing, it's doing its job, and it receives a signal like a growth factor from outside of the cell. So it says, all right, we've got this signal, and it checks, is the cell ready to divide? Is it big enough? Does it have the nutritional needs met in order to divide? If those things have been met, then the cell will move on to S phase. You don't need to memorize the specific cyclin names like cyclin E or B or CDK1. This is a little more detail than you need to know, but you should know that there are 3 checkpoints. You should know where they are, and you should know what are they confirming, right? So then at the end of G2, we have a G2M checkpoint. Like the title says, it's getting ready to move into M phase. And so the DNA has been duplicated during S phase, and at this checkpoint, the cell is saying, has that DNA been duplicated correctly? If something went wrong, you don't want to proceed with bad or damaged DNA, and so the cell will pause at G2 and not proceed into M phase. Similarly, at the spindle checkpoint, the cell checks before it splits the sister chromatids in 2, have the chromosomes all been lined up in the center correctly? If that has not yet happened, the cell shouldn't start pulling that DNA apart. Just going to take a quick minute to talk about these growth factors. So during G1 phase, the cell may receive a signal from its environment to divide. These signals are called growth factors. And one example of a growth factor is platelet-derived growth factor, or PDGF. And you don't need to memorize this growth factor. I just want to show you an example of how it works. In this graph, we can see the growth factor, PDGF, in brown is binding to the surface of the cell, and it's causing all these different, this signaling cascade to occur, all this chain of events. And when you look in the nucleus, you can see that what is happening is ultimately cyclin levels are rising. And we know that cyclin levels must rise for the cell to proceed from one phase to the next. So in this case, the growth factor is ultimately leading to an induction of the cyclins that are required for the cell to enter into S phase. Take some time on your own, again, to watch this animation to help you review the different stages of mitosis and their checkpoints. So let's do a question. At what checkpoint does the cell confirm everything is ready for DNA replication? So to answer this question, you must first think, okay, what phase does DNA replication occur in? DNA replication occurs during S phase. So what phase is before S phase? It is G1. So it would make sense that this checkpoint would be called the G1S checkpoint. It's in G1, it's deciding whether or not it should proceed into S phase and duplicate the DNA. Okay, and here we can see that G1S checkpoint is shown at the bottom in yellow. Lastly, let's talk about cancer in terms of cell cycle control. So the cell cycle is what determines how often cells divide, right? And so if the cell cycle is not properly controlled, cells can divide too often, and that can cause formation of tumors. So ultimately, all cancers are caused by misregulation of the cell cycle. There are two types of genes that can alter the cell cycle. So first, we have tumor suppressor genes, and secondly, we have proto-oncogenes. So these are genes that are important for regulating the cell cycle. But if something happens to them, if they get mutated, they can't do their job well, and so the cell will proceed through the cell cycle when it shouldn't, or it will get stuck in a phase that it shouldn't be stuck in. All right? So normally we like to think of a cell cycle as, in this example, like a race car. And proto-oncogenes are the genes that help tell the cell, yes, let's proceed on to the next phase. And the tumor suppressor genes are those genes that would say, well, we need to slow down a minute. Maybe we're not yet duplicating the done duplicating the DNA, so let's hang out in S phase. Or, oh, we're in mitosis and the DNA has not yet lined up in the center of the cell, so we don't want to start pulling the DNA apart yet. All right? So proto-oncogenes and tumor suppressor genes are good to have, and they are what regulate the cell going through the cell cycle. Proto-oncogenes are like pressing on the gas to move on, continue through the cell cycle from one stage to the next. And the tumor suppressor genes are those that are acting like the brakes to tell the cell to slow down and to stay in its current phase of the cell cycle. Now, if a proto-oncogene gets a mutation, it is now called an oncogene, right? Proto, like prototype. And now this is not a good thing. It's as if the it's as if the gas pedal was stuck down, right? So the cell is continuing through the cell cycle when it shouldn't. It's going round and round. It's dividing over and over again and proliferating too much, causing cancer. So an oncogene is, in our metaphor, is having too much gas. Now reverse, we have our tumor suppressor genes. They are normally like the brakes, all right? In this example, we have, in addition to the brakes, we have this parachute that helps to slow down this race car. So if the brakes aren't working or if this parachute doesn't inflate properly, the cell cannot stop proceeding through the cell cycle, can't slow down, and so again, it's going to over-proliferate. or it may divide when it shouldn't, and it's passing down mutations to the daughter cells, which again can lead to cancer. Let's look at an example of one of these genes called P53. P53 is a gene that's important for regulating the cell cycle. The protein made by this gene functions at the G1S checkpoint. So if you remember, at the G1S checkpoint, this is when the cell has received a signal to divide. It's hanging out in G1, and it needs to decide whether or not it should start to copy its DNA in preparation for cell division. So if the DNA has been damaged in some way, you don't want the cell to divide because you don't want to duplicate the DNA because you'd be duplicating imperfect DNA, and you want the cell to be, and its daughters to be nice and healthy. And so it will monitor the integrity of the DNA, and if P53 sees that there's damaged DNA, it will pause the cell in G1 phase until enzymes can fix that damaged DNA. And if it's bad enough, it will actually tell the cell to undergo apoptosis and die. It's better for the cell to die than to pass on damaged DNA. Now, in many cancer cells, P53 is missing or mutated, right? So, is P53 an oncogene, a proto-oncogene, or a tumor suppressor? Let's think about that for a minute. Here we have normal P53 and we have abnormal P53. Normally, if DNA damage occurs, P53 will sense that and it will recruit DNA repair enzymes. So it will stop cell division. It will stop the cell cycle until this DNA has been fixed. And if it isn't fixable, it will actually just kill the cell. Now, when P53 is abnormal, say it's been mutated, it will not stop the cell cycle at G1, and the DNA that is damaged will be replicated in S phase. So the cell continues on through the cell cycle, and it's dividing and it's passing on this damaged DNA, which can accumulate and cause cancer. So if this gene is normally important for stopping or pausing the cell in the cell cycle, that means that it is a tumor suppressor, right? It's preventing tumors, it's stopping the cell cycle until things get fixed. Next, let's talk about more detail about proto-oncogenes, right? So again, proto-oncogenes, they are genes that are important for the cell cycle regulation. And when they get mutated, then they become oncogenes, and now they are bad for the cell. So proto-oncogenes are good. What they do is they are like gas. So when everything is going good, proto-oncogenes sense that and they tell the cell, we're good to go, let's move on to the next phase of the cell cycle. It's okay to divide. Now, if they get mutated and are acting as oncogenes, now it's as if you have the gas pedal stuck to the floor and the cell is going through the cell cycle, whether or not it should. And so some of these are going to be genes for growth factors. We said that growth factors, genes for receptors for growth factors. So growth factors tell the cell to divide. And so, for example, you could have a receptor that growth factor binds to. If it's mutated, it may tell the cell, hey, it's time to divide, I got a signal, even if no growth factor ever bound to it. Right? So that would be telling the cell to proceed on to S phase when it doesn't need to. Or it could be a mutation in any of those proteins in that chain of signaling. Right? It's lying to the cell and it's saying, we need to divide when it doesn't. Right? And for proto-oncogenes, you only need one copy of it to have a mutation for this to happen. Again, we are diploid, so we have two chromosomes, two copies of each gene, and you only need one of them to be mutated in order to see the effect. Tumor suppressor genes, we talked about ones such as P53. These are the genes that are important for pausing the cell cycle when it's not ready to proceed on to the next stage. Now, if these get mutated, it's like having faulty brakes. However, if only one of the copies of the two genes is mutated, the cell can still has that one good copy, and it is still able to pause the cell when it needs to pause in the cell cycle. However, that cell is now more susceptible to becoming cancerous because it's kind of like, 3 strikes and you're out. Again, we have 2 pairs of chromosomes. That means that we have 2 copies of each gene. So the tumor suppressor gene is present in 2 copies. If you have a mutation that impairs one of them, the other copy is still functional, and so the cell is still able to regulate its passage through the cell cycle, and so you won't necessarily get cancer. However, you are at an increased risk now because there's only one good copy left. Let's look at another example of a tumor suppressor called RB. This stands for retinoblastoma. So this mutation predisposes people to get a cancer of the eye. And it is important for receiving growth factor signals. So it is important in preventing the cell from going on to the next stage until it is ready. So if you have one mutant copy, you can still be okay. The cell cycle is controlled properly, but you're at an increased risk for cancer because if that second mutation occurs, now you have no good RB genes, no good tumor suppressor genes available, and the cell is going to speed on right through the cell cycle, right? The brakes are broken completely, and the cell is going to start to divide when it shouldn't, and you can have cancer occur. Here is a summary of these different types of proto-oncogenes and tumor suppressor genes. You do not need to memorize any of these, including P53 or RB, but you should be able to, if you were given a protein and say, this is what it normally does in the cell cycle, what would happen if it is impaired? This happens when it gets impaired. Is it an oncogene or a tumor suppressor gene? And again, these genes, RAS, SERC, RB, P53, they are mutated in a majority of cancers that we see in people. So these are targets for cancer. So let's finish with the last question. If a cell's proto-oncogenes are mutated and overexpressed, which of the following is most likely to happen? So a proto-oncogene is a gene that's important for telling the cell to proceed through the cell cycle. So it's like the gas pedal. So if you're making too much of the protein, you're telling the cell you're sending too strong of a signal that yes, it's okay, go on and proceed through the cell cycle. And so of these options, A sounds the most correct. The cell will prematurely move to S phase. So perhaps the cell hasn't received a signal to divide, but this proto-oncogene is overexpressed, it's turned on, and it's telling the cell, oh yeah, it's good to go, and it will proceed and through the cell cycle and divide when it's not supposed to. So B would be the opposite, right? The cell would not finish the cell cycle. That's more likely to occur maybe in the case of a tumor suppressor gene. It has no effect on how well DNA is replicated or whether the cell goes into G0 or not. We've never even talked about binary fission, and so the correct answer for this problem is A. All right, again, we have the gas pedal stuck down on the floor. The cell is proceeding through the cell cycle and dividing when it shouldn't.